Good morning, everybody. Thank you for coming to the third annual Synapse Roundtable. My name is Michael Graglia. I am the Managing Director of the Syngap Research Fund, and it is such a thrill to welcome our incredible speaker here today. And over to Dr. Dallman, who is a neuroscientist, I should say, at the University of Miami. And like all of our speakers today, Dr. Dallman is working on a couple of our genes. So in the interest of time, I will let you take it away. Thank you so much for being here.
Thank you so much for inviting me to participate in this roundtable. And I was really, I'm glad I'm following Terry Jo. I use animal models, but we're trying to do this work in the context of also being cognizant of natural history. And so I hope to tell you a little bit about our integrated approach. We've started with Syngap1 and Shank3 models of zebrafish models. Okay, so here's a quick rundown of what I'm going to go through. I'm going to give you a brief introduction, talk about zebrafish models, talk about integrating the clinical and basic studies, and then give a little summary and future direction snapshot. So when as someone who works in an animal model and not even a rodent model, a zebrafish model, we focus on symptoms that we feel like we can make mechanistic insights on, and where the mechanistic insights might inform treatment strategies. And so the two that we've focused on are altered sensory processing and gastrointestinal distress, both I'd say symptoms that we know a little less about. And then one of the things that I've learned in my foray into this field is that the gut itself is an organ that functions through sensing information, nutrients in the lumen, and producing a behavior, peristaltic movements to aid in digestion. So this idea of sensory processing being an important symptom that has big impact on quality of life, this altered sensory processing could play out in the gut as well. And as all of you know, the speed of identification of genetic forms of neurodevelopmental disorders has been very rapid. And so this is just a slide from Satterstrom et al. 2020 that shows some of the single genes of large effect that have been linked to neurodevelopmental symptoms. And I'm circling Shank3 and Syngap. I think I've put in the year in which variants in those genes were linked to neurological conditions in humans. And you can see that Syngap was linked much more recently than Shank3. And as such, the natural history that's known for different conditions is very different, just in part based on when that linkage occurred. Okay, so my lab focuses on these sensorimotor symptoms. And in particular, we've taken a deep dive into GI symptoms, in part because so little is known about how those symptoms arise. And actually, in attending Felon McDermott Family Foundation meeting and talking to families, these GI symptoms were symptoms of big concern. And so that's how we got into looking at these. And so this circle here are molecularly defined forms of ASD with documented GI distress. And this is an incomplete list. Before the meeting this morning, I looked at STX-BP1, and there are also GI symptoms described in that condition, even though it's not listed on this set. Of these genetic forms of neurodevelopmental disorders, 6 have, in animal models, of 6 of these forms have shown that there's reduced motility, reduced movement in the GI tract. And that movement is critical to digestion and gut function because it's part of a reflex that when the gut senses nutrients, it induces these contractions, it induces mucus secretion and hormone secretion that aids in digestion, coordinates with the brain, coordinates behavior. So one of the hypotheses that we're interested in testing is that this reduced motility is a common aspect of ASD-linked GI distress. And then you can see that 3 of these animal models are zebrafish models. I'll show you why zebrafish are useful for looking at GI movement. But there are a lot of genetic models in zebrafish that haven't been examined for GI motility. So zebrafish are a good model for this because by 6 days after fertilization, they have these relatively translucent larvae that are eating on their own. And you can actually watch, this is a movie of a larva that's been fed, and the food includes 6-micron beads. So you can actually watch these peristaltic, the part of the reflex that I was talking about in vivo. And that makes it quite straightforward to quantify what's going on. Different from us, right, where digestion is happening inside and it's not visible. In addition, we've been looking at the response of the nervous system to external stimuli like light. And as I said, we're starting to think about this sensory motor integration, both at levels of gut and brain. Okay, so what have we found? So this is, again, these peristaltic movements that are response to feeding in wild type. This is our Shank3 model, and believe it or not, the movie is going. So there's a little twitch that happens here every now and then. And this is a fed Syngap model larva. You can see that there's actually quite a bit of motility in the gut, but it's not coordinated from a raw anterior to posterior direction. And so we can quantify this behavior, and we can show that, so for instance, in wild type, there's a particular rhythm that is also seen in Syngap, but is slower in Shank3. And so this echoes this need to kind of understand, these studies suggest that, so yes, both are showing GI symptoms, both genes seem linked to these GI symptoms, but manifest in slightly different ways that are important to understand. So the other test that we do in the zebrafish is whole gut transit. And what happens when you, in both Shank3 and Syngap larvae, so what's happening here is we fed them beads in wild type. And in wild type, what happens is the beads are consumed and then make their anterior posterior journey through the GI tract to be expelled. And this usually happens within 12 hours of consumption, the first expulsion. Whereas in Shank3, at this intermediate time, the beads all accumulate at this junction between the intestinal bulb, which is like the stomach, and the upper intestine. And only by 24 hours do you get expulsion. This is true in Syngap as well, and this is quantified here. So what are the cells that mediate this GI reflex? Well, it turns out that your gut is lined, it's sprinkled with these sensory endocrine cells called enteroendocrine cells in light blue. And those actually make most of the serotonin in your body in response to either mechanical or chemical stimulation in the lumen of the GI tract. Those are activated and talk to enteric neurons that activate the smooth muscle cells. So this circuit is really important for taking food in the lumen and inducing the reflex that is mucus secretion and contractility in the gut. And to go deeper into this circuit, we've been collaborating with Liu Huaye and John Rawls at Duke University. The other thing that points us to these enteroendocrine cells as being important is that when we quantify enteric neurons and enteroendocrine cells, which are important for motility, both Syngap and Shank3 mutants have reduced numbers of enteroendocrine cells, which could mean that they're less sensitive to luminal stimuli and therefore don't produce the normal response. And I think these, both from the therapeutic perspective and from animal model studies, most of the attention with these genes that are linked to neurodevelopmental disorders has been focused on the central nervous system. But the gut also has a significant nervous system through the enteric neurons. And, in the collaboration with John Rawls, he does this single cell sequencing of the intestine. And here is, so what they do is they take the intestinal cells and do RNA sequencing on the single cells. And what's shown here are the different types of intestinal cells. And then on this axis are genes that have been linked to these severe neurodevelopmental disorders, and where these genes are expressed in the intestine. And these first 5 rows are the enteroendocrine cells. So you can see that Shank3, catnap, orexin, CDKL5, SCN are all enriched in these sensory endocrine cells, indicating that they could be really functionally important to the GI distress that's impacting these individuals. You can see that other genes linked to ASD are more generally expressed throughout the intestine, and Syngap is not expressed in the intestine of the zebrafish, but is expressed in the enteric neurons. And so this raises the possibility that therapies will have to be cognizant of those cells in the gut that will also need to address the genetics of those cells. Okay, in addition to gut sensory motor integration, we also conducted studies of responses to visual stimuli in our Shank model. And what we find, so this is an assay that is done in zebrafish where you have light to dark transitions, and anytime you turn off the lights, there's a lot more swimming, and that response is dampened in both in 2 different models of Shank3 where the mutations fall either in the N terminus or the C terminus. And what you can see if you map the responses in the brain, the responses to that turning off the lights are that the pineal gland turns on and the hindbrain and spinal cord turn on, corresponding to this increased movement. Whereas in the Shank models, the pineal turns on, but it fails to activate this hindbrain region, which is associated with the movement. So this suggests that the sensory processing deficit is in the hindbrain. So to test that possibility, we transplanted cells from a wild type animal into a Shank3 mutant animal. Here you can see at 1 day post-fertilization, these cells have populated the hindbrain, and this is at 5 days, or actually 6 days when we do the behavioral assays. Now these Shank3 model has a wild type hindbrain, and that actually rescues their response to the light stimuli. So that this shows that the hindbrain is actually, Shank3 in the hindbrain is really crucial, at least in the zebrafish, for responses to light stimuli. Okay, I know I'm losing time, so I wanted to tell you that we're also trying to contribute to better understanding GI symptoms in neurodevelopmental disorders. And one way is, as mentioned by Terry, it's really important to have objective measures. And so it turns out that you can measure whole gut transit time by eating a muffin with a blue dye in it. And this was actually done on an epidemiological scale in England, but not with kids with neurodevelopmental disorders. And we also have, in collaboration with Baharak Moshiri, who's at Atrium Health, we developed a blue muffin that copies the tests that are used that involve X-ray and to measure whole gut transit, with the idea that if we could use the blue dye and look for arrival of a blue poop, that would provide whole gut transit that could be done at home and without intervention. And this is
ready, Sissy?
We have seen whether this is a child with a Syngap mutation, and she's willing to eat
Michael,
it. And
do you
here's
want
another
some more?
child who's
want
being
more?
offered a blue muffin.
Do you like that? Yeah, it's edible, huh?
Okay, so we've also developed an app to try to do, in a prospective manner, gather these symptoms related to GI distress. And what we're hoping to do is use these measures in patient populations at the same time as we're looking for mechanisms of GI phenotypes in zebrafish models to develop strategies for these GI symptoms more generally. And this is the wonderful group that has supported this research. It's been a huge team effort. And so I think I have a teeny amount of time for questions if anyone has them.
Dr. Dallman, do you see the question in the Q&A panel?
Yeah, I do. And I think it is possible that increasing, so there are, you can, yes, this is something that we're planning to test. It's likely that it would work in the model that has problems with the enteroendocrine cells, but we have to test that.